the swabs wouldn't be able to spread infections. But what about the infections that have spread already? How do we cure them? Especially the ones that are life-threatening and affect hundreds of thousands of people each year. One such disease is the well-known acquired immunodeficiency syndrome, or AIDS. AIDS is caused by human immunodeficiency virus, or HIV, and they weaken our immune system. The virus likes to live in bodily fluids like blood. So the transfer of an HIV contaminated blood in a healthy person causes the virus to enter their body and cause infection. Once HIV enters the body, it targets and kills T cells of the immune system. T cells are our soldiers and defend our body against pathogens or harmful microorganisms. The HIV enters the T cells and inserts its DNA into the T cell's DNA. While many of the T cells die, some of them will survive but instead produce more HIV. Over the course of time, the level of T cells decreases and causes AIDS, a condition of weakened immune system that makes a person susceptible to infections and cancers. The available treatment helps to keep the level of HIV low, but there is no definite cure. So the patients have to take medicines for the rest of their life. Sad but true. But Jeff Galvin, CEO and founder, and David Pauza, Chief Science Officer at American Gene Technologies in the USA, thought, if the virus can mess with the DNA of T cells, why can't we do the same? They used gene therapy to edit the DNA of T cells in such a way that they can now resist HIV. If HIV cannot enter and affect T cells, it won't be able to weaken our immune systems and cause AIDS. This has the potential to be the ultimate cure for AIDS, meaning no dependency on medications. Let's hear from Jeff Galvin and David Pauza at the American Gene Technologies about their gene editing technology and how it can save millions of lives.
Imagine a world without HIV. At American Gene Technologies, we think we're on the eve of just that. This year, we may prove that a functional cure is possible and see the end of HIV in our lifetimes. What we've developed is something called a functional cure. If this works, it could mean that HIV-infected individuals could go from lifelong antiretroviral therapy to basically living a normal life.
Our program is putting forth a highly innovative product that hasn't been seen before in the clinical world of HIV medicine. HIV is a unique virus for many reasons. One of the most important is that it attacks T cells. T cells are the drivers of your immune responses, and that means without T cells, you're unable to combat viral infections. So we have to make a change in those cells while we're working with them. And in our case, we put a very tiny amount of DNA into each cell, and that little bit of DNA programs that cell to resist HIV.
And so as a result, those T cells can do their job. They can clear HIV like you clear a cold. We can take HIV T cells and we can make them immune to HIV. Once they're back in your body, you have an immune system that's actually capable of fighting this otherwise very fragile virus. And we believe that's going to lead to a functional cure. An HIV functional cure would be an incredible accomplishment, but believe it or not, it would just be the tip of the iceberg because it's built on a platform of reusable components that you can mix and match to cure a myriad of diseases and that provide a foundation that does 80% of the work of new disease cures in the future.
This milestone is nothing short of a miracle. It shows that we can cure other incurable diseases as well through gene therapy. But you know what is better than a cure? Eradication or elimination of the disease itself. If we could eradicate germs that cause infectious diseases, there would be no diseases to cure. We can say goodbye to infections, symptoms, treatment, and medications. Scientists have been trying to eradicate diseases like malaria, dengue, yellow fever, and Zika fever for decades now. You'll be surprised to know that these diseases aren't